Okay, so good morning, everyone. My name is Tomás Pais and I'm from the Microscopy Facilities team, Nick Barry's team, and I'm going to present today. So first of all, thank you for the invitation to do the talk here, and I'll give the talk about fluorescent labeling in my light sheet microscopy, and we'll have two parts of this presentation. Of course, first of all, fluorescent labeling. We'll start with talking about a bit about immunofluorescence in antibodies. We'll talk then about labeling without antibodies and mainly direct labeling, fusion proteins and tags. And then we'll move on to light sheet microscopy, where we'll talk about a bit about the concept of the light sheet itself, as well as some advantages and uses and things that you can use, samples and types of imaging that you can use with the light sheet. And then we'll talk about two light sheet microscopes that exist here in the facility at the LMB. So first of all, why should you label your biomolecules, proteins, whatever they are, with fluorescence? So first of all, it confers specificity and sensitivity, which means that some of the samples, you might not be able to see them in a microscope if you don't have some kind of labeling. For example, here you have microtube labeling in this muscle, myocyte, so muscle of the heart. On the other hand, you can do multiplexing localization interactions. So you can label many different structures inside one cell, for example. So here you have a dividing cell and you have the chromatin in blue, the mitotic spindle here in red, and the kinetochore in green. And so you can see the different interactions between all of these structures, how they interact, and how their behavior, localization, relation to one another, and all of that. And you can do this also in the tissue. So this is an example of breast cancer section. It has DAPI in blue, cytokeratin in green, vimentin in yellow, and endoplasmatic reticulum in red. And so once again, you can see just the relative localization of all the structures inside the cell. So starting with labeling with antibodies, and we'll start with antibodies just because it's one of the most common and accessible ways of labeling something. And it's basically two techniques: immunohistochemistry for tissues and immunocytochemistry for culture cells, although we basically call it immunofluorescence, and that's just the term for everything. And this basically is labeling using antibodies. So imagine you have a protein that you want to see where it is inside your cell, and you just use an antibody to bind to that protein and to bring it up to localize it. And of course, you have two types generally of antibodies: monoclonal, which target one epitope, and polyclonal, which target many multiple epitopes. And once again, no, sorry, for this one, for the immunofluorescence, you need usually some kind of fixation to connect all to fix all the molecules in their state and preserve the structure, and also some kind of permeabilization so the antibody can actually enter inside the cell. So you have two types of way of doing the immunofluorescence: direct, which you have only a primary antibody that has fluorophores or some kind of fluorescent label attached to the actual antibody, and the antibody just binds to the protein. And indirect, which is also very common, which is having one primary antibody and secondary antibodies that have fluorescence in them, and these bind to the primary antibody. Of course, the advantage of this is that you have signal amplification, so you have more secondary antibodies per primary antibody, so the signal is definitely amplified. And you have also a bit of flexibility, so you have one primary antibody and you can have multiple colors for different. So one day you might label it with red, another day you might label it with green, but the primary is always the same. The direct way is a bit more specific and it's a bit quicker, but it's also more expensive because you cannot use, once again, different colors. As I said before, you need some kind of chemical fixation, and fixation can be generally two types of fixation: organic solvents, and we're talking about alcohols, ethanol, methanol, acetone, all of this. The way to do the way this acts is basically removing lipids in dehydrating cells, and this precipitates the proteins and exposes the epitopes, making the antibody available to bind to these epitopes. On the other hand, we have cross-linkers, and these are things like PFA, paraformaldehyde, glutaraldehyde, all of these, and these basically work as chemically interlinking all of the cellular contents. And basically, in the case of organic solvents, the cell just dehydrates, which means that it shrinks down, which means that the architecture, if you want to see an architecture of a specific cell or structure, it might change with the fixation process. On the other hand, if you're using cross-linkers, you have the cellular architecture remains in the end. Of course, the disadvantage of cross-linkers is that you then need permeabilization. So the antibody enters the cell. Here, the epitopes are, as I said, here are accessible. So yeah, so every technique has its advantages and disadvantages. The idea is basically to use whatever your group, your supervisor is also already using, or if you don't know, or if what the protocol says, or if you have no idea it's a new antibody, try to search for the data sheet, try to search for information, and if there's anything available, there's definitely a bit of trial and error that you need to do. Okay, talking about antibodies themselves. So the antibody is this molecule that has a heavy chain, and this one doesn't change, and has the antigen recognition side on the light chain, and this is the part that changes according to whichever protein it binds to. You can also have the so-called Fab antibody binding fragments, so you can basically cleave the antibody and only use the Fab fragments. And you also have a specific type of antibody, which is the camelids, which actually come from alpacas and llamas and all these animals. And from those, if you cleave them and only have the recognition, the antigen recognition site, you end up with a nanobody, which is much smaller than the whole antibody. And it's very useful to do super resolution microscopy because of this. Okay, so as I said here, the antibodies are labeled with fluorescence. And to have this kind of fluorescence, you have fluorophores, which are molecules that are fluorescent. And the first generation of fluorophores was mainly fluorophores like fluorescein and rhodamine, but of course they had low photostability, low water solubility, so they were not very good at that time. But then you have more modern dyes that were invented and created in the last 15 to 20 years, and these include some of that you already used or already heard about, like the cyanines, the Cy5, the Cy3, and the Alexa fluor series. So you have all kinds of Alexas through covering a long range of the visible spectra. So these are the molecules that will be attached to your antibody and that will make your labeling possible with immunofluorescence. There is a possibility. Imagine that you don't have an antibody that's that has a good affinity to your protein, or for some reason there's no antibody. You can actually use epitope tagging. So an epitope is basically a tag, a tag is an epitope that you can have your cell produce. So you basically have a plasmid vector with your protein of interest, the sequence of your protein of interest, and then you add the sequence of the epitope tag gene, and basically you end up with a protein with a tag in it, and that tag can be targeted by a very specific antibody with very high affinity that you can then use. So there are several types of tags: flag tag, HA tag, V5, MIG, so all of these different ones. And they, as you can see, they target different amino acid sequences here. Okay, so this was basically labeling with antibodies. And I will move on to labeling without antibodies. And we'll talk first about direct protein, directly labeling the protein. So this can, of course, be in various shapes. The first one is actually to have the protein purified. And the idea is that you use an agent. So basically using a functional group that is able to bind covalently to the protein. And examples of these are the FIT-C and the TREAT-C. So some molecules that directly bind to your protein and are fluorescent themselves. Of course, this can be based in kind of in vitro assays where you have the protein itself, or you can just bind in vitro the fluorophore to the protein and then microinject that inside the cell. Okay, so another way of directly binding your molecule of interest are cellular compartment dyes. And once again, these come in various shapes and forms and various various brands sell them and produce them and sell them. And of course, one of the most known is DAPI. It's basically a molecule that is fluorescent and binds to DNA. You can also use HUST, which is kind of an alternative to DAPI. You also have, once again, many of these. So tubulin tracker for tubulin, endoplasmatic ER tracker for endoplasmatic reticulum. You can have the carbocyanide dyes, for example, the di-I, di-O, all of these, and they will bind, for example, in case of di-I, to the plasma membrane, which means that you can actually have your plasma membrane and your outline of your cell labeled. Lyso tracker for lysosomes, fluorescent ceramide analogs for Golgi, and the mito tracker for mitochondria. So the image that was in the beginning of the presentation, well, actually, it's not that, it's one of the samples that we use for training actually has mito tracker, and the fluorescence is still going even after lots of rounds of training. So this is definitely, it stays there. Okay, so you also can take advantage of high affinity natural interactions. So basically some molecules have a higher affinity for specific components of your cells. And all of these here, so are toxins basically that are produced either by snakes or by mushrooms or something, and they can be conjugated with some kind of fluorophore, and you basically use that toxin, put that toxin in your cell, and it binds, for example, here, the alpha BTX conjugates to the synapses, to the acetylcholine receptors. For example, paraclitaxel derivatives, bind to microtubules, and phalloidin, which binds to alpha actin, well, actually to beta actin. And so you can see the cytoskeleton of a cell. And of course, as I said, these are conjugated with fluorophores. So it means that you have a way of directly staining your molecules inside the cell. Okay. Genetic fusion with fluorescent proteins. So once again, we're in the territory of the non-antibodies. So of course, we're talking about the GFPs, and by now, probably you've heard a lot about GFPs, and you'll know that the first green fluorescent protein was cloned from the Aquaria victoria jellyfish. And then from that point onward, you also had the RFP derived from Discosoma. And of course, from all of those, many different wavelengths, many different fluorophores with different fluorescent proteins with different wavelengths were created. And so the idea is that you can produce basically the fluorescent proteins act as fluorescent tags, which means that you can have your cell producing the protein the protein of interest fused to a GFP or to an RFP, which means that you'll be able to localize that protein inside your cell. And of course, this has a bit of genetic engineering, but with tools like CRISPR-Cas9, it's easier to have your cell producing a protein, a fusion protein. Okay, so these also have some variables, some varieties that can be used for different types of experiments. So for example, photoactivatable fluorescent proteins. The idea is here is that you have GFP here, it's just not active. And once you illuminate with a specific wavelength, it actually turns up and then it's active. So you might need that fluorescence to come up in the later stage of development, for example, or a later stage of a cell cycle, for example. You also have photoconvertible fluorescent proteins. So if you illuminate with a specific wavelength, it changes its color from here, from green to red. You also have photoswitchable fluorescent proteins, so you can switch them on and switch them off, once again using different kinds of illumination, different wavelengths of illumination. And then you also have the Iris FP, which is both photoconvertible and photoswitchable. So these are examples of things you can use with specific fluorescent proteins. Okay, there's also one system that's very interesting, which is called the Fushi sensor. And this is basically a cell cycling labeling. So these are a few constructs that you can buy, and then you can inject them in a cell. And the idea is that you will have a specific fluorescence depending on the phase of the cell cycle where you are. So these are based on ubiquitination of these two cell cycle regulators, geminin and CDT1, which means that if you're in the G1 cycle, you're basically the color that will appear is red because it's geminine that's going to be ubiquitinated. And then the opposite, if you're in G2, it's CDT1 that's ubiquitinated, and so you only see green. When you're in S, basically none of them are ubiquitinated, which means that you'll have green plus red, which is yellow. So this is a system that you can just use for your cells and just see in which phase of the cell cycle you are. Okay, there's also this thing called mini-SOG. So this is basically mini singlet oxygen generator. And the idea is for this one is that it's a label that can be used for fluorescence, for light microscopy, and that can later be used for electron microscopy. So it's good for correlative fluorescence and electron microscopy. The idea is that this protein tag here basically will catalyze a reaction of DAB, which is a staining that can be used for electron microscopy. So wherever you have the green, the green there, the fluorescence there, you'll have also DAB staining later on after the process of DAB staining. So this is good once again for correlative in electron microscopy. Okay, you also have genetic encoded non-fluorescent tags. So the idea here is that you had the tags and then you had, for example, an antibody binding to the tag. Here, or the fluorescent protein being fused to the protein, or the fluorescence, the fluorophore just binding directly to the protein. Here, the idea is that you have the fluorescent probe or the fluorophore just binding to a tag there, either without the need for an enzyme to catalyze the binding, the reaction of the binding, or with an enzyme that just does that. So, for example, Flash and Reash from InVitroGen is a system that you have this tag over here. If you have this tag without the fluorophore being added, it's non-fluorescent, and it's only when the fluorophore binds to the tag that you have fluorescence and that you see your cell. On the other hand, you also have these kinds of tags, the snap, the clip, and the halo. And it's basically, once again, a tag that you have a compound here that binds to it, and then you produce fluorescence. So you can have different types of tags and then bind these different compounds and have different structures being labeled inside a cell. So for example, here you have a Drosophila neuron, Drosophila brain, and with two different colors. Okay, so ACP and MCP tags are basically what I explained before. So these are tags that you also have for fluorophore binding, but in this case, you need the ACP or SFP synthase to catalyze this binding reaction. Okay, so just to do a summary of what we've been talking so far about labeling, you can do multiplexing with fluorescence labeling. So you can have, the idea of labeling is not only to identify a specific structure inside your cell or a specific cell, but also to identify different structures or different cells. You have immunofluorescence conferring specificity and sensitivity on fixed samples. Choosing the fixation method, of course, is important, depending on whatever you want. And also you have other techniques like fluorescent protein fusion for live imaging, also some rapid chemical labeling. So for example, for those labels followed in all of that bind directly to your molecules. Okay, so we're going to move now to light sheet microscopy. And to explain a bit what the concept of light sheet microscopy is, we go back a bit to wide field and confocal. And so in wide field, you have all your sample being illuminated, and all the light that's coming from your sample goes to the camera or to the detection system. In confocal, you have also all of the sample being illuminated, but you only get the light that's coming from a specific focal plane. All of the rest of the light gets rejected, and it's only that light that gets to the detector. And so you have an optical section of your sample. What happens in light sheet is that you only illuminate a specific focal plane. So here in both of them, all of the sample is illuminated, which of course can create photobleaching because you're basically imaging only this plane and then this plane and then this plane, but you're illuminating all of the sample as you are illuminating all of the planes. But here, the idea is that you use a specific cylindrical lens to actually illuminate your sample and produce a sheet of light that is just illuminating, so only the focal plane of the sample that you want. Okay, and then you have a detection objective that can just collect the illumination coming from your sample. Okay, so that's what I said, only the focal plane is illuminated, and this produces the optical sectioning, and of course, this reduces photobleaching because all of the sample here and here, the areas of the sample that are not being illuminated, they're not being bleached, so that fluorescence can just keep there, and then once the focal plane moves there, then they will be illuminated and the image will be acquired. Of course, nowadays, the first light sheet was just that, a light sheet that did light sheet imaging, but nowadays there are lots and lots of different microscopes that use light sheet illumination. So we actually will say more that it's the type of illumination that is light sheet instead of the microscope, the type of microscope that is a light sheet or not. And, but generally speaking, for all of these light sheet systems, there is one concept that is very important, which is that because the light sheet length and the light sheet thickness, so the thickness of the actual sheet of illumination, depends on the numerical aperture of the objective. This means that you cannot have increasing one without the other. So if you have an increase, if you want to have a less thick, so a thinner light sheet to do a thinner optical section, then you end up with also a shorter light sheet length. Okay, so you always have to have a compromise between choosing thickness and choosing length of your light sheet. And once again, this is generally for all of these types of light sheet microscopes. Okay, so how to mount a sample for a light sheet microscope? It's a bit different. So you're used to having a normal inverted microscope or an upright microscope, and you just put your sample on a cover glass or on a slide. Here it's a bit different because you have one objective that's being used for illumination and another objective that's being used for detection, either being like this or other types of configuration. So you cannot put a cover glass here or a slide here. And so the idea is that one of the most common ways of mounting a sample is actually having these tubes here. So these low melting agarose or just normal agarose tubes, they are produced using these capillaries. So you just have an agarose solution inside the capillary solidifies. And before it solidifies, you can just put your sample there. And imagine that you want to image a Drosophila embryo or a zebrafish embryo, or a big chunk of tissue. You can just place it there, and there you have your sample. You just dip it in the light sheet chamber over there. It's suspended here, and then you can just image it and illuminate it with the two objectives. So it's a different way of mounting a sample. It might need a bit of optimization depending on the sample, but if you're using things like Drosophila and zebrafish, once again, many people already did this, so it's actually getting easier and easier to do. Okay, so just a few examples and a few reasons for using light sheet. So this is a Drosophila embryo, and as you can see, this is being imaged, and this is a 5-minute continuous illumination. So we have 30 frames per second, so we have 10,000 frames, and as you can see, the time lapse is still going. It's sped up in the middle, so it's a bit faster here, but there's no photo bleaching there. So if you would have a confocal, basically you would have all of this being illuminated with the point scanner all the time. So this would photo bleach much faster than the fluorescence is going here. Okay, there is, however, an issue, which is scattering. So if you're imaging a large volume, a sample that has a large volume, as you realize here, so this is a Z stack. This would be the result of a Z stack here. And this would be where the objective would be. And the image starts to be very crisp, but at some point, light starts to scatter. And then you end up with a blurry image there. And this is exactly the same. So this is crisper than this one here. So how to solve this scattering problem specifically in using a light sheet microscope? The idea is that you can actually, using that sample or that way of mounting a sample that I showed you before, you can actually rotate that cylinder, rotate that tube, and have the acquisition being done through different angles there. So it means that you can have acquisition being done here, and then the sample rotates 90 degrees. So sample is here, acquisition here, rotates 90 degrees, acquisition rotates 90 degrees, which means that you're actually acquiring from this plane and then from this plane and then from this plane, for example, which means that you solve a bit of the problem of scattering. And then the idea is that you can use, different kinds of software, for example, Fiji or Huygens, to actually reconstruct all of these different acquisitions that you have from different angles. Okay, so to explain a bit how this works, so this is a case where you have Drosophila salivary glands here, and these were mounted for the light sheet using also beads inside the cylinder there. And so these beads are used basically for kind of having a registry of the position and also the rotation. And this is the fusion of all, this is a time lapse. So this is the mounting, and this is a time lapse experiment where you can see the salivary glands developing. There they are. So this is the fusion of all time points, but just from one angle. And then you can have a rendering of all of 3D of all the time points. So this is from all of the different angles, and then this is the 3D reconstruction. So as you can see, you have less scattering, and the image is much more uniform through all the planes there. Okay, so another example is a fixed organoid, and this is being imaged in the Zeiss Z1, which is a commercially available light sheet microscope. And as you can see, of course, you don't have, this doesn't solve scattering in the core of your sample if it's a very thick one, but it certainly solves it in the periphery, in the outside of your sample. And otherwise, if you would be doing illumination and image acquisition only from one side, you could not see. So you could see the image being crisp just here and this side, and it would be blurry on this side. And then you have crisp image all around your sample. So this definitely improves in terms of scattering. Another example, mouse oocytes here. And as you can see, once again, it's very uniform throughout. Okay, this is done by Kate McDowell. This is a mouse embryo developing. And as you can see, this is just lots of time points. And in this case, you actually have another a way to speed up, or not speed up, to make image acquisition a bit faster, which is to actually use, instead of just one detection objective, using two, and then you can acquire images a bit faster. And as you can see, this is the gastrulating embryo. We can just see it from different angles, and once again, very crisp all over. Okay, another configuration that you can use light sheet is this, is for cultured cells. So you can have this, the idea, of course, is that once again, you have the illumination and the detection objectives, and they have this, they once again have this angle here. They're just placed closer, and you can have the cover slip there. And then you can use this for fast and high resolution imaging. And just to have an idea, these are human cells, and one volume is acquired every 7 seconds. And once again, the image is still going, the fluorescence is still going, so it's very good for bleaching. And then you have also neurons here. Okay, so now in terms of what systems do we have available here at the LMB in terms of light sheet? So we have the subcellular light sheet and the oblique plane light sheet, which I'll just explain in a moment. And then we have some other systems that are either bought by specifically by some of the PIs, or are also custom built by them. For example, the one that I just showed you from Kate McDowell is the SimView. You can talk to her if you want to use it. And we'll start with the subcellular light sheet. So this is one of those that you can use. This is built by James Manton. And this is one of those where you can use that configuration where you can use cells. So you can use a cover glass. And the idea here is that these are dictyostelium, and using confocal microscopy, these would bleach. They're very sensitive, so they would bleach very, very fast. So you wouldn't be able to have all of this time of acquisition, and there's still fluorescence going on. So this is the main advantage, definitely, or one of the main advantages of using light sheet there. So diffuse thallium sample, and it would bleach very fast. So this is another microscope that we have, another light sheet, which is the oblique plane microscope. So as I told you here, you have this configuration there, but if you want to have a very thin focal plane, the problem is that if you're acquiring the image, of course you have the objective being obliquely oriented toward your sample, which means that the focal plane has an angle that's not the same as the angle where your cells are, where your monolayer of cells are, for example. And so to solve this, basically this oblique plane microscopy was created. And the idea is that you have one objective here, and that objective is acquiring this image with a specific angle, and this image is kind of distorted. And then you have another objective here that's producing an image from the image that this one produced. And then you have this remote image over here, and it's also demagnifying it. So you basically have an inverse of whatever you have here with the same size of the original sample. So the advantage now is that you can place a third objective here, a third microscope, that this one you can actually place with a specific orientation that would counter for the orientation of your cells, and to actually have it with the focal plane being the same orientation as the cells are. And so this one will image this one here, and then you end up with a correct image with the correct focal plane. And this was also built by James Manton with help from Jacob and from Akash, and you can talk to them also to use this microscope. Okay, so a little bit of a summary about light sheet. There are definitely many ways to do light sheet imaging, and the idea is that it mainly reduces the phototoxicity, so photo bleaching, that's kind of the main advantage. And of course, you have multiple imaging, which allows to solve this problem of the scattering. And there are these kind of custom built 2 options for imaging at the LMB with the systems that can belong to the facility, but you can also talk to some of the PIs that have their own light sheets in case you want to use them. And basically this was it. If you have any questions, I'll be happy to answer them. Thank you very much.